Hello everybody, thank you very much for having me, and thank you for allowing me to speak in English, which is better, I think, than Spanish, probably, which is the other language that I know. I'd like to talk today about phase-amplitude coupling and some work that we're doing at HMS to use this in a variety of domains. The rough outline is that I'm going to be talking in a little bit of a general way about rhythms and coupling in mostly neural data. Then I'm going to talk about phase-amplitude coupling and some of the ways that this is measured, and a recent project where we use PAC to illuminate some of the neural circuitry of arousal, and then some of the other ongoing and future projects using PAC that we're working on. If you saw Hukun's talk this morning, he discussed how there are rhythms in physiological and neural data at a variety of scales, including scales around a day, having periods around a day, such as circadian fluctuations. There are ultradian rhythms with periods around an hour, and then there are rhythms such as Meyer waves having periods of several seconds. I'm mostly going to be talking about faster oscillations in EEG activity. So traditionally there are about 5 bands that EEG activity has been divided into, because there are robust rhythms in these bands that have been observed in a variety of different conditions important for cognitive functions. I'm going to be focusing today on 2 or maybe 3 bands, depending on who you ask. Theta rhythms, which occur at frequencies between 4 and 10 hertz, and gamma rhythms, which classically occur at frequencies between 30 and 100 hertz, although recently they've also been observed at higher frequencies between 100 and 200 hertz. So these rhythms are not just epiphenomena. It's been hypothesized that they represent oscillations. So they represent the fluctuations in the activity of neuronal populations, with the size of the population in inverse proportion to the frequency of the rhythm. There's a great book that you can read if you want an introduction to this topic. So they also are associated with a variety of cognitive processes. They have real cognitive importance. So, for example, gamma waves appear to be related to encoding of stimuli and representing features in the brain. For example, visual stimuli are associated with robust gamma frequency rhythms in the visual cortex. Theta rhythms are associated with working memory and sequence recall, as we'll see. Now, these rhythms are recorded at different sites on the brain, and coupling between rhythms appearing at different sites also has cognitive significance. For example, the phase locking of gamma rhythms at, for example, the visual and auditory cortices may represent the binding of stimulus properties, such as visual and auditory properties of a stimulus. And phase locking between theta rhythms in, for example, the hippocampus and the prefrontal cortex may represent the transfer of information from memory stores to executive centers. Recently, a lot of attention has focused on coupling not only within frequencies, but across frequencies. And there's a little cartoon here to kind of indicate just how this coupling occurs, and there are a variety of ways. So here's a low frequency amplitude modulated signal, and here are several high frequency signals, and the phase, amplitude, and frequencies of these signals are related to properties of the low frequency signal. So here we have a high frequency signal whose amplitude is modulated identically to the amplitude of the low frequency signal. Here we have a high frequency signal phase locked to the low frequency signal. Here we have a high frequency signal whose frequency depends on the phase of the low frequency signal. And finally, we have an example of a high frequency signal where the amplitude of the signal is modulated so that it's highest at certain phases of the low frequency signal. So I hope that's clear. This kind of cross-frequency coupling is most robust and was first discovered between theta and gamma frequencies. And it turns out that both the phase and the amplitude of gamma rhythms are often locked to theta phase. So I want to give you guys a quick and I think very compelling example of how this kind of cross-frequency coupling can be used as a coding scheme in the brain, which is working memory and remembering sequence of digits. So I want you to think about remembering a phone number. So phone numbers both here and in the US, I believe, are 7 numbers. And there's a good reason for that, because the average working memory capacity of a person is 7 digits. There's also data that indicates that when people are rehearsing like a phone number to themselves, they do it at a rate of about 7 numbers per second. So if you're trying to remember a phone number, you repeat it in your head about once a second. So there's a very interesting set of hypotheses that have been developed to explain these observations about working memory in terms of the nesting of gamma oscillations within theta cycles. So this is an example of phase coupling between theta and gamma, as well as phase-amplitude coupling. So the idea is basically that each digit in a phone number is represented by a different neuronal population. These should be numbers, not letters, but and that these neuronal populations fire roughly at gamma frequency. When you are remembering something like a phone number, what you do is you take, so you have gamma cycles nested within a theta cycle, and on each gamma cycle within the theta cycle, a different neural population representing a different digit is activated. Now, this accounts for why the capacity of working memory is about 7 digits, because if your gamma rhythm is about 60 hertz and your theta rhythm is about 8 hertz, then you have seven gamma cycles within each theta cycle. So here you can see the 7, well actually here it's about 5, well about 7 gamma cycles nested within each of these theta cycles here. Okay. Furthermore, when you are mentally rehearsing something like a phone number, it may be the case that the number that you rehearsed is that the neuronal ensemble representing it is activated at the peak phase of the theta rhythm on sequential theta cycles, which accounts for the speed of silent rehearsal. Okay. So I think that's kind of cool. So that brings us to phase-amplitude coupling. And before I talk about phase-amplitude coupling, I just want to make sure that we're all on the same page about what phase and amplitude are. So here are a variety of signals in black, with phases indicated in blue, and amplitude indicated in red. So here is a constant sinusoidal signal. Its amplitude is constant, indicated by this constant red trace. And its frequency is also constant, so we get a phase that just increases linearly, with 0 phase corresponding to the peak of the oscillation. Okay? Here's an example of a signal which has constant frequency, so again we get this very regular phase trace in blue, but its amplitude is modulated, so we have this kind of irregular red trace. Here's an example of a signal with roughly constant amplitude, but decreasing frequency, and you can see that the phase is no longer linear, but it decreases sort of like a square root. You can also have signals with amplitude and frequency modulation, such as this more complex signal down here, and those are the kinds of things that we'll be looking at in brain signals. So, okay, so this brings us to phase-amplitude coupling. So phase-amplitude coupling occurs when you have a signal, like this simulated signal here, with 2 oscillatory components. A slow oscillatory component, such as a theta rhythm, and a fast oscillatory component, such as a gamma rhythm. Now, the phase-amplitude coupling comes in because the fast component has an amplitude which is modulated in phase with the slow component. So that's very easy to see in this cartoon of a simple signal, but you can also see it in a real, this is a local field potential recorded from the brain of a mouse. Here's the raw trace, and here is a theta filtered, this is the theta filtered trace. This is a trace filtered at low gamma between about 40 and 80 hertz, and this is a trace filtered at high gamma between about 110 and 180 hertz. So you can see, especially in the high gamma trace, that there are bouts of high amplitude on roughly the descending phases of the theta rhythm. Now, sometimes that amplitude is higher than other times, and sometimes there's high amplitude on other phases, but statistically the relationship is that the highest amplitudes occur on the falling phases of theta. So, phase amplitude coupling has been found not only between theta and gamma oscillations in the hippocampus, but in a variety of brain areas, and now it's very widespread in neuroscience. It's also different from spectral analyses like the bispectrum or bicoherence, and it's different from other coupling measures like phase coupling, as I pointed out earlier. PAC has already been used to uncover rhythmic phenomena that you can't see in the spectrum, and I'll show you an example of that a little later on. And it's been functionally linked to all kinds of cognitive processes, including perception, memory, and decision-making, some of which you'll see later. Finally, there are some very interesting general hypotheses about how phase-amplitude coupling may be involved in the coordination of neural activity across scales. Because, as I mentioned, high-frequency oscillations are associated with activity of small populations of neurons, whereas low-frequency oscillations are associated with activities of large populations of neurons. So when you have interactions between low and high frequencies, you may have, these may be accompanying interactions between large and small populations of neurons. Okay. So I want to show you guys how phase-amplitude coupling is measured most often, and then I'll show you some results on phase-amplitude coupling. So the first step is to take your EEG and filter it. And there are a variety of ways to do this. In this example, we've used convolution with a Morlet wavelet to get a variety of theta traces at frequencies between, center frequencies between 4 and 12 hertz, and a variety of gamma traces at center frequencies between 20 and 180 hertz. Once we have two rhythms that we're interested in calculating the phase-amplitude coupling between, what we want to determine is whether the amplitude of the high-frequency rhythm depends in a statistical way on the phase of the low-frequency rhythm. So we extract the phase of the low-frequency rhythm and the amplitude envelope of the high-frequency rhythm, and then we bin the amplitudes according to the phase of the low-frequency rhythm. So all the red points here are phases around pi of this 9 hertz rhythm. The red points down here are the corresponding amplitudes of the 60 hertz rhythm. When these are all added up, we get an average amplitude. We get average amplitudes represented by these red bars here. These green points represent phases close to 0 of the 9 hertz rhythm, and the green points here represent amplitudes of the 60 hertz rhythm at those time points. When those amplitudes are all added up, we get average amplitudes represented by these green points here, these green bars. So this distribution of amplitude by phase shows us that in this rhythm, even though it may only be a statistical relationship, there's higher amplitude on average at around the phase of pi. Okay? So this gives us an indication of whether there's phase-amplitude coupling between these two rhythms. To go a step further, we try to quantify that relationship by determining whether the phase-amplitude distribution is uniform or not. So we use something called a modulation index, which is a measure of the inverse entropy of this distribution. If you don't know what that means, it basically determines how far this is from uniform. So here are 4 example signals where In the first case, there's almost no phase-amplitude coupling, and in the last case, there's quite a bit of phase-amplitude coupling, which you can see in the amplitude by phase distributions. These are the modulation indices corresponding to these 4 cases, and you can see the modulation index is 0 for this nearly uniform distribution of amplitude by phase, but it's higher for this clearly non-uniform distribution of amplitude by phase. Okay? So we take this distribution of amplitude by phase and we get a number out of it. And to get a comodulogram, we basically repeat this process for a variety of frequencies. So on this axis, we have frequencies between 4 and 12 hertz. We had about 40 filtered traces at these frequencies and 40 filtered traces at these frequencies. These small traces here are the amplitude by phase distributions for each pair of signals. And we quantify the uniformity of these and get a comodulogram. Here you can see there's very non-uniform distributions of amplitude by phase, and those correspond to high modulation indices indicated by hot colors. Okay? All right, so hopefully I haven't lost everybody yet. The last step is to remove spurious indications. So you can see here that sometimes there's non-uniformity, but it's not due to a clear relationship between phase and amplitude. It's basically due to noise or other aspects of the signal. So we'd like to remove that, and we do that using a surrogate data technique where we shuffle high and low frequencies between different epochs. So here you can see the raw modulation index calculated for EEG from mice during wakefulness. Here is the modulation index calculated when we shuffle gamma and theta between epochs. And subtract off this background coupling, we get an indication of where there's significant coupling between about 6 hertz and about 85 hertz. Okay, great. So now that you guys know what this comodulogram is, let me show you a couple of results showing some of the cognitive significance of PAC. So first I want to show you results from rats. There's a very cute rat over here running a T-maze task. So this rat would like to get this cheese at the end of one of the arms of this maze, but he doesn't know where the cheese is until he hears a tone in the middle of the maze, and the tone tells him whether to turn left or right to get the cheese. So he's trained to recognize the tone. Here are comodulograms calculated from 30-second epochs from each of the different sections of the maze. So you can see this is comodulograms calculated from local field potentials recorded from the striatum, which is involved in decision-making and reward processes, and from the hippocampus. So let's look at the hippocampus first, actually. So in the hippocampus, you see very dramatically that there's theta-gamma coupling exactly when the rat is hearing the tone. And at that point in time, he's trying to remember whether the tone is telling him to move left or right. So we might guess that this theta-gamma coupling is indicating that memory process. This is a little less clear, but there's high coupling between theta and gamma frequencies in the striatum during the whole time the mouse is waiting to hear the tone. So presumably he's attending, thinking he's going to get a reward, something like that. Okay. This occurs not only in rats, but also in humans. Here is data recorded from epileptic patients, the hippocampuses of epileptic patients during a working memory task. And you can see that the coupling between theta and gamma frequencies is much greater during working memory tasks than during baseline. And also that the preferred frequency of theta to which gamma is coupled decreases with working memory load, which may have something to do with the fact that when you are trying to remember more numbers or more objects, you need a larger theta cycle in which to nest more gamma cycles. Okay. Finally, here's a very interesting and very recent result that a lot of our work is based on, showing that during REM sleep, coupling between theta phase and the amplitude of both low gamma and high gamma is much higher than it is during active waking, quiet waking, or non-REM states. So there's this preferential sort of biomarker of REM sleep, which is theta gamma pack, and people don't yet understand fully the significance of that. So I'm going to show you a project that we've been using this result to kind of illuminate some of the neural circuitry of arousal. Okay, so the parabrachial nucleus is a brainstem nucleus that basically is involved in taste perception, and people also think it's involved in arousal. And our collaborator Satvinder Kaur from Beth Israel Medical Center has tried to probe this by activating the parabrachial nucleus using genetic technology, using a DREAD designer drug, which activates a designer receptor. But she found that when she activated the parabrachial nucleus, her mice were exhibiting a state where they weren't moving a whole lot, but would respond to stimuli, and she sort of couldn't tell whether they were actually awake or asleep. Furthermore, when she looked at local field potential, EEG recorded from these mice, she couldn't use standard sleep scoring techniques or spectral techniques to determine whether they were awake or asleep. So just to show you sort of what that looks like, here are average power spectra from an hour's worth of each of these states, REM, non-REM, wake, and a parabrachial nucleus activated state. And you can see that there are sort of distinctive spectral markers of REM and wake that you might expect. So there's high theta activity as well as low and high gamma activity during REM. During wake, you have high theta, high low gamma, and then you have a broad sort of a bunch of power at higher frequencies. When the parabrachial nucleus is activated, you get something that could sort of go either way. So you have high theta power, you have low gamma power, you have a bump of high gamma power, and you also have this sort of broad increase in higher frequencies. If anything, it looks from this like the parabrachial nucleus activated state is more like the REM state than the wake state. Now, what we did is we took this data and we calculated comodulograms for an hour's worth of data in all these states, including the first hour of parabrachial nucleus activation, and then hours 4.5 to 5.5, hour 8, hour 11.5 to 12.5, and the last hour about 15 hours after the parabrachial nucleus was activated. So what you can see here is, first, that our results are in agreement with already published results regarding REM, showing robust coupling between about 7 or 8 hertz and both low gamma and high gamma. That's stronger than any coupling seen in non-REM or wake. But we also see a new result, which is that the coupling during wake, in contrast to published results, is not at the same frequencies as the coupling during REM. And we think this may be due to the fact that published results are from parietal cortex, where this data is from frontal cortex. Now, if we go on and look at what happens when the PBN is activated, we can see that there's a very clear indication that, at least to me, that the PBN activated state is more similar to the waking state than to the REM state. The frequencies of phase-amplitude coupling are nearly identical between waking and the PBN activated state. And that persists during almost the whole time of PBN activation, although there is some change as we get to the end of the 15 hours. If we go a step further and look at the amplitudes of various gamma-filtered oscillations according to theta phase, we get supporting evidence for all these conclusions. We see that REM is stronger than non-REM in wake, but unlike previously published results, the phase at which gamma is coupled to theta during wake is different from the phase at which gamma is coupled to theta during REM. And by looking at the profiles, the amplitude by phase profiles of the dread state, we can see that it's much more similar to wake than to REM. So in conclusion, this PAC analysis allowed us to see what spectral analyses couldn't, which is that the activation of the parabrachial nucleus induces a state which is much more like wakefulness than like REM sleep. We also see a novel result, which is that in the prefrontal cortex at least, the preferred theta phase and frequency are different during wake than they are during REM sleep. Okay. I don't know, how much more time do I have?
You still have 50.
Oh, great. Okay. Okay, so let me talk fairly briefly about some of our other projects involving phase-amplitude coupling. So we've been looking at, hand in hand with our study of parabrachial nucleus activation, we've looked at the effects of different filtering approaches on phase-amplitude coupling quantification. So most people use narrowband filters like the Butterworth filter at order 3 to calculate comodulograms. We tried broadband filtering techniques, including the empirical mode decomposition and serial dyadic filter bank, which has filter response properties similar to the EMD and similar to the wavelet transform. Now we tested these 4 approaches on short simulated signals having a 65 hertz amplitude modulated component coupled to a 6 hertz phase modulating component. So this, in order to get, because these are broadband filtering techniques, in order to get accurate frequency estimation, we had to use a local in time frequency estimation, and then use that to basically figure out exactly what frequencies PAC was occurring between. But we found for a wide variety of artifacts that we introduced to our simulated signals that the broadband filtering techniques yielded much crisper frequency resolution in terms of PAC between 6 and 65 hertz. And that narrowband filtering techniques led to very poor resolution in frequency, or in fact spurious assignment of phase amplitude coupling between frequencies where it wasn't actually occurring, as you can see there. Okay, so we've also tried to employ PACs to look at signals at frequencies much lower than those seen normally in brain activity. So we looked at multi-unit neural activity of the circadian clock and searched for PAC at frequencies from about we had periods of about 2 days through periods of about half an hour. And we found, as we hoped or expected to find, we found very robust coupling between daily rhythms in activity and ultradian rhythms having 5-hour or about 2 or 3 hour periods. We found slight differences between animals so this is in mice also. We found slight differences between animals in alternating 12-hour light and 12-hour dark cycles, and between animals in constant darkness. There seemed to be stronger coupling in the light-dark cycles. And we also looked for coupling between ultradian rhythms, having periods of about 6 hours to 40 hours, and rhythms, 6 hours to 40 seconds, and slightly higher rhythms. Unfortunately, or maybe fortunately, we found sort of an interesting result here, which is that the comodulograms we discovered were very sensitive on the surrogate data techniques that we used. So here we have 5 different surrogate data techniques, and we get almost completely different palettes of coupling given those different surrogate data techniques. So this is something we're trying to explore further to understand if there's a systematic way that these comodulogrammes depend on surrogate data techniques, and what is the optimal surrogate data technique for a given signal and a given pair of frequencies. We're also trying to understand the relationship, or the cognitive significance of the PAC that's seen very robustly during REM. So REM sleep is known to be important for certain kinds of learning, for consolidating certain kinds of memory, especially emotional memories. And we are now looking at data taken from mice trained on the Morris water maze, which is a task that has been shown to exhibit REM-dependent learning, and looking at how the magnitude of PAC changes over multiple days. In between each day, the mouse is trained further on the Morris water maze. So you can see here, there's remarkable robustness in terms of the coupling frequencies exhibited across days, but there also seems to be some fluctuation in terms of the intensity of coupling. And the very preliminary results indicate that the proportion of REM sleep is inverse to the amount of coupling seen during REM sleep. We are in the middle of trying to assess, trying to sort of verify this result, but this is kind of interesting. And finally, we're trying to take a more dynamic approach to PAC quantification. Most people look at average phase-amplitude coupling across long periods of time, hours or longer. But this phase-amplitude coupling, we hypothesize that it's not constant, it's transient. So we'd like to determine when it shows up and why. And we also want to see whether it occurs between brain regions, between theta rhythms in one brain region and gamma rhythms in another brain region, and exactly what is the network of PAC that is observed between brain regions. We're also looking at how PAC is perturbed by different pharmacological agents and different genetic manipulations. We have a lot of data to work on. And I think that is about it. I want to thank my collaborators, Kun Hu and Wan Xin and Min Song, as well as our experimental collaborators from Beth Israel Deaconess, Savinder Kaur, Heinrich Gomp, Bernat Coxis. I have to thank the funding agencies, and I also want to thank my hosts at the conference and all of you for listening to my talk.
Thank you, fabulous presentation from Polletta. Is there any comment?
Question. So it seems that the comodulogram is kind of a standard way of measuring proteins. Can you tell us a little bit more? Because I have some difficulty reading the axis, what the axis at phase given period or whatever.
Sure.
And if they seem to have different scales, I think that the scale means at least the frequency span.
Yeah, that's right.
How to read it?
Yeah.
Sure. So you guys have software to do it also.
Yes, we do.
Later on, I'll talk to you.
Yeah. So basically, what we have down here is, this is the frequency of the low frequency oscillation.
Okay.
And the way that this works is, what most people do is they kind of they select a number of candidate low frequency oscillations and a number of candidate high frequency oscillations. And they compute the degree of coupling between all of those, every pair of low frequency and high frequency oscillations. And that's what's exhibited here in this color map. What you see then is you see that for some pairs of candidate oscillations, there's very little actual coupling. While for other pairs of candidate oscillations, there's high coupling. So there's some indication of between which frequencies you really see coupling and where there isn't any coupling. We're actually working on a little bit of a different approach where the frequencies here are not based on candidate oscillations. They are derived from these oscillations on a cycle-by-cycle basis.
Another follow-up question is, you showing both this pattern between theta and gamma.
Yes.
Are they attempts to try other frequency bands?
Yes, yes. So in fact, that was the thing I was going to mention is you can choose any sort of, so from our software anyway, you can choose any span of frequencies here and here. And for example, in some of our results, we've looked at much broader scales, like these are sort of circadian scales. In brain activity, there's also phase amplitude coupling between delta and theta, alpha and beta, beta and gamma, theta and beta. It's all over the place. And the interesting thing is teasing out the different contributions of these couplings to different cognitive tasks.
It seems to me that you're implying that the fast is somehow usually driven by this slope.
Yeah, so
But is there any significant feedback? And also, how far are you guys from actually modeling the dynamics?
So that's a good question, and clearly the answer basically depends on the model that you're using. There isn't really directionality in phase amplitude coupling. There's just an association between the amplitude of a high frequency rhythm and the phase of a low frequency rhythm. Many of the models of theta gamma coupling that are currently existing indicate that the causality goes in the direction of theta modulating gamma, so that it really is causal from low to high frequencies. But in other domains, for example, phase amplitude coupling has been seen in geological signals, in financial signals. We're seeing some of it in physiological signals. I think all of those will have to be taken on a case-by-case basis, whether the causality is from high frequency to low frequency or low frequency to high frequency, or whether there's another mechanism which influences both simultaneously.
Yeah, so it's very possible that third frequency will come into play, serving as a barometer of the system.
Yeah, definitely. Yes.
Thank you.
Okay, I'm sorry to tell you that due to the deviation time, we have to finish the session. Thank you for all your attention.